How are you guys? Let's complete section 8.2, structure of the DNA. The last thing we talked about was the nucleotide structure, and we know now there are 4 nucleotides, and those 4 nucleotides are thymine, adenine, cytosine, and guanine. Now my question is, if I told you that for a long time scientists hypothesized that DNA was made up of equal amount of the 4 nucleotides, what's your opinion? Is this hypothesis true? Do you think that all organisms have the same proportion and equal amount of the 4 nucleotides? If so, why do we have different instructions? Why do we carry different traits? So of course, this hypothesis wasn't correct. After that, another scientist called Chergaff started to analyze the DNA to find out the proportion of ATGC as the 4 nucleotides. He found that the proportion of A equals to T and the proportion of G equals to C. And he called this Chergaff's rule. Chergaff's rule stated that the amount of adenine is equal to the amount of thymine and the amount of guanine equals to the amount of cytosine. But actually, did you know anything about the arrangement of those nucleotides? Do you have any idea about the shape of the DNA? How are those nucleotides arranged? Why they are similar in proportion? Why A equals to T and G equals to C? Actually, he failed to explain that, and he failed to put a 3D structure for the DNA. After Chergaff's rule, there are 2 scientists started to realize that since A equals to T and C equals to G, so of course the DNA is in the form of double helix. So the 2 scientists, Watson and Crick, started to build a model. This model, 3-dimensional model to figure out the structure of the DNA in the form of double helix. And they said there is phosphate group and sugar. They put the phosphate group and sugar as a backbone outside the model. Outside means in the outer area of the double helix. Double helix, so there is inside the 2 helixes and outside. Inside the 2 strands and outside. Outside the phosphate group and sugar, inside bases. Another scientist started also to study the structure of the DNA. This scientist called Franklin. Here is Franklin. Franklin started to use X-rays crystallography to take an image for the DNA, and this was the image. What's your opinion? Is it in the form of double helix? Yes. So this X-ray crystallography or this X-ray image supported the idea of Watson and Crick for the DNA in the form of a double helix. So now Franklin's X-ray image suggested that the DNA was in the form of double helix. Not only that, but with even width. What do you think the cause of this even width? Why the DNA has the same width all over its length? Because actually, according to Chergaff's rule, A equals to T and C equals to G, so of course there is pairing between A and T and pairing between C and G. Look, They called this idea the base pairing rule. This is the base pairing rule. A pairs with T, C pairs with G. If you notice that in each pair there is one pyrimidine and one purine. What does it mean? In each pair there are 3 rings. That's why there is equal width all over the length of the DNA. That's why Franklin found the even width of the DNA, or the even width of the double helix, because look, G and C, 3 rings, purine with pyrimidine. A and T also, purine with pyrimidine. That's why the DNA has equal width. And this is the structure of the DNA. In the model of Watson and Crick, they said that nitrogenous bases inside the double helix between the 2 strands. Look, A, T, C, G. What's outside the phosphate group with the sugar? Here it is. Phosphate group with the sugar. Which bond can bind all those nucleotides together? Which bond can bind the sugar with the phosphate group? The bond is called covalent bond. Covalent bond to bind the sugar with phosphate group. Which bond can bind G with C, A with T? Hydrogen bonding. Hydrogen bonding. So inside the model, or inside the double helix, hydrogen bonding to bind the nitrogenous bases, the paired nitrogenous bases, and covalent bond to bind each phosphate group to the sugar. This is the structure of the DNA. Thank you for listening. Bye bye.